Welcome to Cancer Lesson 3.5, How Do Cancer Cells Evolve? The goals of this lesson are to explore how tumors contain different populations of cells with different mutations, and consider how eliminating a tumor will require treating each population differently. At the end of the lesson, the students should be able to understand the sequence of driver mutations required to transform a cell into a tumor, define the terms monoclonal tumor and polyclonal tumor and explain their relevance in cancer progression, and interpret data showing that chemotherapy can select for drug-resistant tumor cells. We will achieve our goals by engaging in an interrupted case study exploring how the heterogeneity of a tumor affects our ability to treat it. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. Cells can hyperproliferate once they have acquired mutations in 3 types of driver genes. A tumor with only one pattern of driver mutations is called a monoclonal tumor. Different genes can act as drivers, so cells can have different patterns of mutations. This kind of tumor is polyclonal. Treating a polyclonal tumor effectively requires dealing with all the different cell types. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure. The student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet. The key points of the do now are that cells need mutations in all 3 classes of driver genes, tumor suppressors, proto-oncogenes, and telomerase, or ALT, in order to hyperproliferate and become immortal. Mutations are random and there are many different kinds of driver genes, therefore transformed cells may have different mutations. Most tumors consist of populations of cells with different mutations, and so they are polyclonal. The do now begins by asking students for examples of key classes of driver genes that need to mutate in order for a cell to transform. The driver mutations need to inactivate tumor suppressors, increasing survival, activate telomerase, preventing senescence, and activate oncogenes, increasing proliferation. Then the cell will transform into an immortal, hyperproliferating cell and form a tumor. Exposure to carcinogens increases mutation rate, increasing the likelihood that the key drivers will mutate and transformation will occur. More than one gene can behave as a tumor suppressor or an oncogene. There is even an alternative to telomerase, ALT. So different cells may take different paths to transformation and immortality, as long as the 3 classes of genes are mutated. As we begin to understand how many genes can behave as drivers in each class, it isn't surprising that most tumors are polyclonal, or heterogeneous. In fact, the different cell types in a tumor compete with each other for survival. The key points of this lesson's activity are that because cells in a tumor have mutations in different genes, they may need different drugs to kill them. Cells that are not killed by a drug will continue to proliferate, and this is called drug resistance. Drug resistance in tumor cells is therefore analogous to antibiotic resistance in bacteria. We'll get there by having students complete an interrupted case study on susceptibility and resistance to chemotherapy. In this lesson's activity, students will work in groups examining how the polyclonality or heterogeneity of tumors will make them differently susceptible to chemotherapies. Students will also explore how a chemotherapy can actually select for a resistant population of cells within a tumor. The wrap-up reinforces the point that if a tumor contains several types of cells, a treatment that eliminates only one type of cell will allow the other cells to carry on proliferating and take over the tumor. To eliminate this tumor, we will have to use a different drug that attacks these cells. Much like how antibiotics select for resistant bacteria, so chemotherapies can select for resistant tumor cells. The fact that a polyclonal tumor may contain a multitude of cell types makes them even harder to eliminate. The selection may not even result in cells that grow better. They may grow slowly but spread better. Either way, the tumor persists. For homework, have students read an article from the Daily Telegraph about resistance to chemotherapy. One of the most common challenges students have is that the term driver genes in this lesson is different from the drivers of proliferation, proto-oncogenes, in Lesson 2.2. We apologize that the field uses this term in two different contexts. In Lesson 2.2, students learned about one type of driver gene, a proto-oncogene. This lesson expands the concept of the driver to all types of genes that push the cell towards cancer. Therefore, the definition of driver genes includes key genes that are involved in cell survival, proliferation, aging, and invasiveness. At the end of the lesson, collect the lesson worksheets to assess students' explanations of why polyclonal tumors are difficult to treat. The challenge that tumor heterogeneity presents for designing effective treatments will be explored in the next unit. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.